owners and their custodianship of the lands on which we meet. We will pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognise their valuable contributions to Australian and global society. Just some housekeeping. Today's seminar is scheduled from 12 to 1 o'clock. We may not take all this time, but there will be plenty of time for questions. We ask that you utilise the question and answer tab, which is available on the Zoom functionality. We will not be monitoring the chat, so if you do have a question, please use the question and answer function, and we will facilitate these questions at the end of the seminar today. We do ask you that you hold your questions. By all means, use this functionality during the seminar, but we'll be opening up for questions at the end of both seminars from Bronte and Tim. I'm now going to hand over to Michael, who will introduce Bronte and will commence the seminar. Thank you for joining.
Thank you, Kieran. It gives me great pleasure to introduce our first speaker, Dr. Bronte Sutton. Bronte is a graduate of Charles Sturt University, Wagga Veterinary School, and she brings to us today a wealth of practical coalface experience from across the entire livestock supply chain, both in Australia and in Southeast Asia. And she has great experience in the delivery of animal health, welfare management programs, and is going to be able to talk to us today about her perspective, an industry's perspective, on the potential impacts of lumpy skin disease in Australia. So without further ado, I'd like to introduce Bronte to present her seminar. Thank you, Bronte.
Great, thank you. I presume you can hear me and see the presentation okay. So thanks very much for having me today. It's a real honour, and I'm really pleased. I definitely don't have all the answers about LSD. Unfortunately, none of us do, but I think the best way to move forward is definitely for industry and, government and research partners to all move forward together, because, it is obviously a very alarming time, but equally there is some, really good science and forward-moving progression that we can do together that is going to change the game in terms of LSD that hasn't been available to us previously. So introducing AgTrade, for those of you who are unaware of it, AgTrade's a global livestock genetics and food security business. So we have a very wide suite of kind of subsidiaries, and that includes domestic breeding, growing and feedlotting, both dairy and beef cattle, domestic and international, boxed protein, global livestock and feed, including a big footprint in the hay space, and including hay export. And probably what you know us the most for is AUSTREX, our exporting branch, and that's a global livestock exporter, working mostly with cattle, and exporting from Australia, New Zealand and South America, and the bulk of that is going to Indonesia and China. My history, as Michael's already said, is 8 years with AgTrade, including several years living and working in Southeast Asia, and most of that time was spent in Laos, Cambodia and Vietnam, with large-scale dairy and beef operations there. And I now in my role back home as group veterinarian for AgTrade, I oversee the kind of animal health and welfare opportunities, but also the challenges across our diverse network on a day-to-day basis, as well as our kind of strategic directions in those areas. And now I'm happy to be home and also owning and running my own rural livestock veterinary practice in rural New South off our small family farm. So I guess as a company, but also as an individual, we're in a bit of a unique position to apply some of our learnings in, I guess, biosecurity and market resilience, and particularly specifically in LSD, but also FMD and other diseases from our kind of global experience, and wondering how we can best leverage that to help in an Australian context. So moving straight on to LSD specifically. I know you're all very aware of it, and I'm sure very around the mechanics of the disease itself, but some of the kind of quick recap on the most salient points from a strategy perspective. LSD has been spreading through the Middle East and Greater Asia. I mean, really since 2012 is where it really started to get a run on, where it's previously just been percolating away as an endemic disease in Africa. But particularly in the last 4 years is when it's been, moving south through Southeast Asia in a very big way. And Indonesia announced their incursion in March 2022, so this year, and it's slowly been spreading south in that time. Obviously, with then layering FMD on top, LSD took a little bit of a back burner in terms of management and operations and media in Indo, and that has its own unique effects in terms of awareness, management, and reporting. Our 5-year modelling of incursion risk is up to 28% this year after the Indo announcement. That's back up from 8%, 12 months previous. And obviously an incursion into Australia would affect the cattle and buffalo exports, particularly live and particularly major product derivatives, and reducing the value of the Australian cattle industry. What would that cost? No one that I know of has been game to put a figure on it. The FMD outbreak has been estimated at 80 billion over 10 years in terms of potential cost to Australia. The 2011 class action was worth 2 billion, and that was only a 6-month ban on live export. So if you extrapolate that, that's about 40 billion. That was only live export. It's really hard to know, but it would be safe to say it'd be significant, and I would estimate it would be up around an FMD incursion. There is currently no vaccines registered in Australia, and we know there's inherent issues with this. Previously in other countries' experiences is obviously problems with live vaccines and their cleanliness in terms of biosecurity and having other unwanted diseases mixed in with it. Return to virulence of those vaccine strains, as well as there being so much LSD percolating now. There's an unprecedented drive for vaccines. So there's been significant shortages, and in an Indonesian context, this has definitely held up LSD vaccine availability that is only really now, becoming widely spread available. It's really important to know that no outbreaks have been controlled without vaccination anywhere in the world, and no eradications have been successful without the use of a vaccine. So this kind of comes comes up again in one of our key threats. So let's talk about some of the key threats and then move on to key opportunities. Obviously, the timeliness of identification and confirming an incursion will be key to how far that incursion is spread by the time we realise, and therefore how we can appropriately respond in accordance to the AUSTREX plan's guides. And what our experience will be that's unique to other countries is definitely the limited population, the geographic isolation, and the limited kind of animal monitoring on a day-to-day in the kind of the vast north. Another thing we need to consider is the ongoing presence in neighbouring countries year on year, as Indonesia is really, looking towards endemic nature at this stage. So even post an initial incursion and management of that, what it looks like kind of year on year risk. I mean, a lot of countries that have been successful in controlling outbreaks or eradicating the disease, because of the big effect of kind of seasonal impact, weather patterns and vector action, they can have free status for many years and then see re-emergence, several years later. Other issues for us is obviously the multiple insect vectors, including potential of novel vectors. And we're all very aware of the role that ticks, cattle ticks and other ticks, could start to play in this. Another part is our trading partner response. So both their real response, that if we had an incursion and that would, or what products that there would be a breach about their health certificates for export, as well as any kind of opportunistic plays on market access or market price that would surround that. Health and welfare management in a northern context goes back to that geography and isolation, and how do you support animals like that during such a extensive northern scenario. And then what has really been a real challenge, definitely for Indonesia, and even we can see it in Australia, like concurrent exotic disease threats, so FMD and Japanese encephalitis, and all of that percolating together impacts the resources, the personnel, as well as kind of industries' appetite or ability or headspace to respond. And that's the same for the public. So we need to bear that in mind when we target stakeholders for education and responses. And then, of course, as we really saw as FMD hit barley, and the service and disservices that media does for us, the market volatility that surrounds these things, and that all being around, buyer confidence. The good news is that there's a lot of opportunities to work together to get, the best outcomes possible. We really need to empower all stakeholders to recognise and respond. And there's been some really good work done in this space by a lot of different areas. But one of our key learnings from Indo was that definitely everyone needed to be involved. You couldn't afford for, some areas of the supply chain to be left, so it's every part of the breeder, grower, abattoir, as well as all the kind of ancillary services and public around that. Good relationships with our trading partners is key, really, when we get into the need to renegotiate our health certificates for exports, and really to reduce the intangible fallouts of kind of mistrust or opportunistic kind of market pushes. Vector control, we're in the unique opportunity that we can learn off the what we've learned from Japanese encephalitis and what the pig guys have really brought to the table lately, as well as some novel technologies in vector control and some lateral applications. So we've been working on that in Indonesia about, fly tags and things like that, and do they have a role in trying to manage things? Remembering that these are mechanical vectors only. So in fact, they're messy eaters and it's in their mouth parts, but only transiently. So at least that means the overwintering doesn't occur as it does when they actually become a biological host in their own right. But most importantly, it's about rapid diagnosis, and how do we do that in a extensive northern context, as well as having access to a safe, effective vaccination that does not affect our OIE freedom status and is practical in an extensive northern context. and that doesn't exist. But what I really want to celebrate and thank is the people who are doing so much work into this, and we can't give them enough support because they're the people who are going to save us as an industry. So I think that really leads straight into Tim's talk, please. Thank you.
Thank you very much, Bronte, for setting the scene for our next presentation from Professor Tim Mahony. It gives me great pleasure to introduce my colleague, who he describes himself as a molecular virologist, but Tim is an innovator, and in particular looking at developing innovative approaches to immunising livestock against endemic and exotic diseases. So without further ado, I'd like to invite Tim to present to us today his seminar, which is entitled Research Update on Lumpy Skin Disease. Thank you very much, Tim.
Thank you, Michael, for that very kind introduction. It's always a pleasure working with you. I'd also acknowledge the traditional owners on the land on which we meet today, and acknowledge their ancestors and descendants. I'd like to thank Bronte for a really excellent introduction into the problem of lumpy skin disease that we will be talking about today. And I guess the good thing is that some of the stuff that Bronte had in her slides, you're going to see me reiterate that as we go through the talk today. A little bit of an introduction. I mean, I guess as Bronte indicated, one of the issues that we're really facing at the moment is it's not quite a perfect storm in terms of exotic diseases, but it's certainly getting there. FMD has been on our radar forever, really, and we have a lot of systems and things in place to try and prevent that from entering our country. I guess earlier this year, we were surprised somewhat, I don't think Bronte was, that lumpy skin disease turned up in Indonesia, and then soon after it was followed by FMD. Now, for our producers, that really, as I said, is a clear and present danger, in that we really, while we prepare for these things, we really don't want them to. I'll talk about those two a little bit. What are the risks that might drive that, particularly with respect to lumpy skin? Bronte also touched on some of those. And then I'll talk about some of the approaches we might use for developing the vaccine for lumpy skin disease. Okay, Bronte again touched on this point. I guess it might be surprised for some people to know that endemic diseases are a big problem for our industries as well. So it's estimated that around over $3 billion is lost annually to that. But it's really the exotic diseases could have that huge impact. Now, I think my figure is the same as Bronte's, which is always good when the speakers agree on the same day, on the same seminar. FMD estimated around $60 billion over 10 years. One of the big impacts of that will be potential livestock standstill across the country. And we've seen what happens to the human population when that happens over the last couple of years during the COVID pandemic. Once we have that, then it's a lot of work and a lot of time to regain our FMD-free status so we can get access to those markets again. Now that's both virological in terms of controlling that outbreak and then demonstrating our freedom from disease, but then we have to regain those access to those markets that have probably been taken up by some of our key competitors. So it's not just as simple as getting rid of the virus. So we'll compare the two viruses just briefly. So for those of you who are interested in virology, as I do myself, they're quite different viruses, so belong to different. LSD is a pox virus. Again, we have the emergence or re-emergence of monkeypox throughout the world at the moment, so they're closely related to that. FMD is a coronavirus. Not much to say about that, except that it has influences on their physical characteristics, which can be important when you're trying to decontaminate a particular environment. They have different genomes, Why is that important? Pox viruses have a double-stranded DNA genome similar to us. The importance of that is that they tend to be more stable than RNA viruses, which FMD has, although for an RNA virus, it is relatively stable. Airborne transmission, unlikely with LSDV, although you look at that, and we'll talk about that in a moment. FMD is well recognised to be a highly airborne virus with quite widespread transmission from infected animals. Mechanical transmission, by what I mean that is transfer of the virus from, say, infected clothing or dirt on boots, that sort of thing. Very unlikely for LSDV, but yes, it's quite common for FMDV. Insect transmission, as Bronte indicated, yes, definitely yes for LSD. That's the main mode of transmission of this virus. FMD, it's a sort of a putative yes in that it's not a key mode of transmission of the virus. Host range, buffalo and cattle for LSDV were all cloven hoof for FMDV, so therefore a potentially wider impact, not just in the cattle industries, but also the pig industry as well, and sheep also could be very important there. Environmental persistence, we would not expect LSDV to persist in the environment, whereas FMD is well documented to do that. And again, animal persistence, an animal gets infected, it recovers from the infection as far as we can tell, but then the viruses may still reside within the animal, and there's potential for that then to drive an outbreak further down the track. In terms of our local interest, we've never had an LSDV incursion on Australian soil that we know about, whereas, and the last one for FMD was 150 years ago. And even that, at that time, generated a very prompt response to try and eradicate the virus and also track down where it might have come from. So even that long ago, it was still considered a very important virus. So as we've talked about today, FMD is currently, according to the official status at the moment is that it's under control in Indonesia. But what is our potential risk to Australia? So before the pandemic, I was surprised that there's so many flights to Bali, and apparently over 1.2 million people visited Bali in Australians visited Bali in 2019. So that's a lot of people. And from that perspective, we would think that people are the most likely vector to bring FMD back into the country. And we've seen a lot about that in the media and from our politicians in the last couple of months since that incursion was in that. The other potential source also via people is undeclared food. So we know that's always a huge problem with our borders. But what's on our side is that we know that the potential likely points of entrance. So it's going to be international airports. So we know if people are going to be bringing that in, that if we monitor those, then we have that potential to have that intervention. And we've seen enhanced awareness for our politicians and the media, and then increase in biosecurity measures at our international airports. The other key point with FMDV, if we can get access to them, we have highly developed and highly effective vaccines for FMDV and diagnostics to complement those. So because of that well-known historical risk of FMDV to many countries that are free of the disease, we have this underpinning scientific knowledge. Now, if you go into PubMed and put in FMDV, you get a return of about 3,000, just over 3,100 publications. I'd be interested to know if anyone could guess what the figure is in brackets of nearly 300,000. So FMD is the oldest known animal virus, was first described in the late 1800s, about 1895. And so over that the subsequent 100 years, we've had just over 3,000. So the one that's almost 300,000 is COVID-19. So in the last 2 years, going on to 3 years, there's been nearly 300,000 publications just on that virus alone, with that disease alone. A bit of an imbalance there. If we look at LSDV, we know that there's been reported cattle infected with the virus in Sumatra. It's a long way to Australia, so about 3,000 kilometres if we go from somewhere in the middle of Sumatra to Broome on the northwestern coast. And we would think that the most likely vector for an LSDV incursion is to be an insect host. We have systems in place in our airports and ports to monitor for the potential accidental importation of potential insects because of these types of reasons, although we know that it does happen. So we've seen incursions of the red fire ant, for example, and so it always remains a risk. The other one is climate and weather. So on one of Bronte's diagrams, we saw that the prevailing winds come from that direction onto our northern coast. And we also, and so what's not on our side with regard to LSDV is we had a strong focus on LSDV. We then had the outbreaks of FMDV, and LSDV didn't fall off the radar from an industry point of view, but from other aspects it has. So it hasn't received the same sort of publicity or awareness around it. And I think the other thing with LSDV is we really don't know where that entry point might be. So it could theoretically be, and if it was to come in via it's unlikely that these insects may come in through our airport. So if they were blown in from the north, then we really don't know when that might occur. An animal might get infected, and then the virus might move to a susceptible host in that particular area, and so it could be there for quite a while before we know about it. So perhaps, and again, I'm glad to see Bronte and I really agreeing today, one of the key things is there's no widely available suitable vaccine, and similarly, there's no widely available or suitable diagnostics to support the use of those vaccines in controlling an outbreak. I think that's good. And then when we go down to, well, what's the base knowledge of us, we know about LSDV, we go to PubMed again, there's about 260 publications of that compared to just over 3,000 for FMDV. So our basic knowledge base on LSDV is clearly a lot lower for LSDV. So what's the risk in LSDV? A bit further information on that. So Bronte again articulated this very well. We've seen it move across Asia over the last 2 to 3 years. And you can see on this diagram here, 2019, just in the eastern parts of between India and China. And then we move on earlier this year, and it's moved down into Sumatra. And so this is this rapid spread, and it really is rapid when we think about that for the probably 100 or so years we've known about LSDV. It was largely restricted to Africa, and it's only really started that move fairly recently from the virus point of view. So this is this key risk that we face, and how do we manage this and trying to stop it reaching our northern coast. Fortunately, from our point of view, we do have some models in place that might enable us to really understand this risk a bit better. So we have the National Arbovirus Monitoring Program, which looks at diseases which are of importance for live export. So our partners might require that the cattle or other animals are free of particular diseases. And so these arboviruses are ones that are also insect-borne by LSDV. And so sometimes when we're modeling these viruses, what we're actually doing is we're modeling the hosts or the vectors that might actually transmit them. The key one is bovine ephemeral fever, or 3-day sickness for some. High cost to industry annually. It's an endemic virus, so it's always present in the country. We have a live viral vaccine for it, which is used by industry. There are some, we haven't got time to go into that today, but there are some thoughts that vaccine could be better. But it moves from the north to the south annually, or more or less annually. And it's also transmitted by mosquitoes and kilopoides, which I thought potential key vectors for LSDV. So bovine ephemeral fever provides us a potential good model to see or to think about what might happen if we were to get an LSDV incursion in this part of Australia. And as I said before, it essentially follows the wet season and the consequence of vector breeding but it's restricted from moving too far south because of the vectors. So the vectors can't survive there because of cold temperatures, and that's essentially where bovine ephemeral fever stops. And that might give us some thoughts about, okay, that sounds pretty good. We'll have, but the weather can control that for us. But of course, as Bronte indicated, there may be other vectors that we're not aware about for LSTV that may not actually work for ephemeral fever. And then if we look more closely at, and so this is to show that when we look at it, if we had an incursion of LSTV over here somewhere, that the darker, as the colour goes into it, these darker browny reddy colours, and the increased cattle population. So there's nothing, this is how ephemeral fever moves, and we see that there's really a strong line of available hosts that move down the coast, but it's restricted by the vector, not the actual availability of hosts. So we can't be complacent just by relying on the weather, as we should know. And so these are some examples of bovine ephemeral fever spread over a reasonable length of time. As you can see, that on an annual basis, how far south the virus might move varies a lot, and that's presumably, I didn't have a chance to go into it, but it's presumably related to things like rainfall and warmer weather, that sort of thing. And when we see more or less classical, if you like, transmission or movement of bovine ephemeral fever, this might be what we consider to be a normal climactic season. But as we know, we don't really know what normal climactic means these days. Whereas in some of these other examples, when we can see the virus really doesn't here, it's actually in 2910, it's actually just reached the northern parts of Victoria down on the Murray. So we really need, just relying on climate alone or this potential change in vector survivability, that we can't rely on that. So we still need available technology. So as I said before, it's a long way from Sumatra to the northern coast of Australia, about 3,000 kilometres, but we still think the most likely vector is going to be an insect, perhaps through our airports or ports. Climate, again, as we all know, very hot topic across the globe at the moment. It's more variable, so we can't rely on it like we might have once done to try and keep this sort of thing out. I guess the current risk level could be described as be alert but not alarmed in terms of LSTV because of this. There was some work done by Peter Durr at CSIRO in Geelong where they developed a tool to model the spread of or potential spread of vectors that might transmit virus using their TAPIS tool. And they were looking at a blue tongue virus, so that's part of the National Arbovirus Monitoring Program because it's spread by the Chilocoides type biting midges. And what Peters did in this study is his co-authors, they reviewed using their modelling tool historical cases of what were thought to be wind-spawn bred of the blue tongue virus. And what they discovered in that study here, and you can see this up here, that they were able to show that the potential spread of the vector, so the Chilocoides biting midges for the blue tongue virus, is probably limited to around about 150 kilobases. So there's molecular virology coming through there, Michael. 150 kilometres are not thousands of kilometres. But like Australia, Indonesia is a big country. So currently the virus sits over here, but in Indonesia extends over here, where it's only hundreds of kilometres from the northern tip of Australia. So again, it shows, and I think, Bronte raised that point, that we just can't be complacent about this. The current physical barriers, such as geography and things like weather, are going to protect us, and we really need to be preparing our own strategies to deal with this virus if it was to come into the country. And then we can think about, this was something I didn't know about, and consider this. So the desert or dust storms from the Sahara Desert, they are actually responsible for blowing dust across to Caribbean and northern South America, so to the Amazon rainforest. And so if dust can be spread far, then it's not inconceivable that an insect might be spread over a longer period of distance that we just haven't documented it to date. And these types of storms happen sporadically, so it's not a one in, all in. But in this case here in 2018, I think it was, there was actually, June 2018, there was a continuous dust cloud from Sahara Desert across the Atlantic to North and South America. So again, that potential, that change in how our climate might be behaving could change the game in terms of what happens with these vectors. So I have to move on now to talking about what happens in an outbreak, what might happen in the UK, and Michael's well aware of this. He was in the UK in 2001. There was an outbreak just under a year, cost over £8 billion. That's a lot of Australian dollars. And in that outbreak, there was just over 2,000 confirmed cases. Over 10 million animals were culled. And the reason for that is that thing about trying to get back to disease control and then get back to disease-free status. So here is an outbreak in America in the early 1900s, I think, from memory, early 20th century. And this is what happened in 2001. So over that 100 years, we didn't learn very much. The same things happening. But apart from the potential, there is an ethical and logistical dilemma in trying to dispose of these large animals over this short period of time. Around about the same period, there was an outbreak in the Netherlands. Again, a low number of cases. They did use vaccination, but then there was nearly over 250,000 animals slaughtered to try and get back to that disease-free status. And I guess one of the key things, and Bronte touched on that, is even if we have a vaccine that's available, we still need to be able to tell which animals are being vaccinated and which animals are being infected and or vaccinated. So the ones that we're after don't always stand out from the crowd. So we have to develop the complementary diagnostics to allow that to happen. And so this is a concept which is referred to in vaccinology and animal science of differentiating infected and vaccinated animals, or DIVA. And essentially it relies on animals which have been infected having different responses or different measurable immune responses to those which have been vaccinated. And so in this case here, we see that the animal has been infected, the red one, and it's developed antibodies to the virus and then with the structural parts of the virus, but not to the non-structural virus parts of the virus. So those things involve replication. Whereas in a vaccine virus, because the virus doesn't replicate, these components are missing. So the animal, when it gets vaccinated, only generates to the structural parts of the virus. And so we can use these types of approaches to try and develop complementary diagnostic tests that enable us to differentiate these two classes of animals. And so when we're talking about using vaccination to control an outbreak of zoonotic disease, it's crucial because we have to be able to absolutely say that this animal was vaccinated, not infected, otherwise we don't get back to that disease-free status. Bronte mentioned the current LSD vaccine, which is the Nephilim strain. It's a modified live, so it actually infects the animals. And she also mentioned, which time I won't go through this, but I guess the key thing with the LSDV, apart from reversion of virulence, is there's no capacity for DIVA. So if we use the Nephilim strain, then we would not be able to differentiate those infected and vaccinated animals. Now we've developed, looked at developing live viral vaccines for a DIVA type approach. And in our approach, what we've proposed in the past is actually adding in a novel antigen. So something that doesn't necessarily or doesn't normally occur in that particular virus. And so when you immunize with it, it raises a response to that. And we know if there's responses to our marker antigen, it could be something like GFP, so green fluorescent protein from jellyfish. If they have antibodies to that, then it's unlikely that they must have been vaccinated because tails don't get attacked by jellyfish very often. So when we look at the LSDV, what we've been doing and what our plan for this year has been to try and develop a vaccine for lumpy skin disease, but incorporating that DIVA capacity so we can tell which animals were vaccinated and which animals weren't. So what we've done is we've gone through the LSDV genome. So this is a pictograph of that here. It's complex from the virus point of view, really interesting viruses, and I could probably talk for a long time about that. From that point of view, not today. Identified those structural elements that we need animals to respond to that, and then produce immune responses that will help protect them. So we all have heard about mRNA vaccines in the last couple of years. So we could potentially make an mRNA vaccine for LSDV by identifying, so these coloured boxes are the genes from the LSDV genome, and then express them in various formats to try and then deliver that to animals and then have them respond to that. We could incorporate our novel antigen, if you like, from say GFP from jellyfish again, and we could look at doing various strategies to do that to try and develop our novel vaccine. However, there's a group in New South Wales that are already developing an mRNA vaccine or looking to develop that for LSDV. So what we want to do is try and complement that. So we can't have all of our eggs in one basket, but we don't all want to be doing the same thing as well. So what we've done is we're going to developing a subunit DIVA vaccine for LSDV. And what we're doing at the moment is we're expressing 4 antigens that we've identified from LSDV that we think should be able to provide protective responses. And then we've also developed what we call our synthetic antigen. So this is something where we, rather than using something from a different organism, we've actually taken components of the LSD virus and put them all together in a way in which they don't exist in nature. And so if animals have responses, immune responses to that, then we know that they are going to be, that they've been vaccinated and not infected. Now, by using LSDV components to do that, I think that's a good way to go, because then there's no way that we're going to interfere with the diagnostics of other things. So if we can't use FMD, for example, because then if we deliver our vaccine, then we wouldn't be able to say that we complicate those FMD diagnostics. So where are we at with this? We're down here, so we're working really closely with our great colleagues at DAF, and at UQ we're looking at expressing these antigens in yeast, and then we'll use those to formulate into our vaccine. DAF at the same time are developing complementary. So as I said before, we need those complementary technologies to complement our vaccines so that we can improve the protection of the virus in the field, but then also that differentiation of infected versus vaccinated animals. So we're various ways along with the various antigens in terms of expression for the vaccine, and Beth Fowler and the group at DAF have been developing molecular assays to detect the virus itself, and then we'll also express antigens in insect cells to develop to be able to detect antibody base. I'll leave Beth to talk about that another day. And so we hope that we will be in a position to be able to formulate and test our prototype vaccine in the model species, but ideally in something a bit larger, such as a pig, somewhere towards the end of this year. And then we can theoretically be ready for testing in adults. So why is our subunit vaccine so novel? So what we want to do is we want to incorporate that into our single dose technology. So a couple of years ago, we developed a single dose cattle tick vaccine. Now, as Bronte said at the start of the seminar, if we're going to use it in the north, we have to be able to use it within the limits of current industry practices. So in the south, where we have a much higher density of cattle, vaccination programs are arguably easier to roll out. Whereas in the north, if you're only nestling once or twice a year, and if you've got a multi-dose conventional type vaccine, then that's not going to be very useful, is it? Because you're only seeing them once or twice a year, and you might need to deliver doses once a couple of months apart. So in our cattle tick vaccine, which is a technical term, is artisan microcapsulated formulation, but that's for the polymer chemists to worry about. You can see here this graph depicts the antibody responses in cattle immunised with it and a conventional double dose. So they've got one here and one here. Double dose. These animals, we've only given them one dose at day 0, and you can see that while the responses are variable within group, they're persisting much higher than we see in our conventional dose. When we did cattle tick challenge studies with these, the efficacy, they were challenged 2 months after vaccination. The efficacy for our polymer formulation was equivalent to them being vaccinated twice, but only a couple of weeks after that second or third vaccination. So we think that if we can formulate our LSDV vaccine in the same way, it will be highly useful for using that northern industry. So in summary, I think we just have to keep it aware, keep it on the radar. Both of these viruses are critically important to keep them out of our country. We probably have most of the systems and vaccines that we require for FMD. LSDV, we don't, and we really need those to stop the spread. Stopping it in Indonesia is critical, because even though we might be able to use these on shore, we will still have those economic impacts as a consequence of that. If we can keep it, help Indonesia control it in Indonesia, then that is obviously our best option. Just quickly, I'd like to acknowledge the team at DAF and Mike at UQ that are going through these processes in the expression of the various antigens for the vaccines and the diagnostics, and Mohammed and David from Queensland University of Technology in Melbourne, who were the chemists who developed that formulation. Thank you very much. Over to you, Michael.
Thank you very much, Bronte and Tim, for 2 excellent presentations. Now we have time for questions. So at present, we only have 1 question. So I'll give people time to, using the Q&A function, to add in their questions. But I'll start off with firstly a question to both of you, and Bronte, you might start, and then Tim, you could follow. My first question is, from what we know today, what are the major reasons for the spread of the lumpy skin virus from Middle East through Southeast Asia, and what's contributed to the fairly quick spread? Do we have a good understanding of that?
I mean, I don't think we have all the answers on that at all, but it's been a significant, volatility in weather, and therefore, vector activity. And, for similar reasons that we've seen Japanese encephalitis in Australia and never seen that before, or why for a number of years we've seen bigger and bigger BEF, like 3-day results. So it's, I don't know if you want to blame climate change, how you feel on that, but I will certainly put it up there.
Thanks, Brian. I think you describe it as climate variability, maybe slightly less controversial then, perhaps. Yeah, I tend to agree. I mean, I don't think we really know, Michael. I think I think we'd have to go with that. I mean, I guess there's been some recurrent outbreaks in Israel that I was reading about. They tried to track down, and it seems that they thought they might have been vector, flying of vectors in, but it turns out it was probably more likely to be illegal movement of animals. So probably a whole combination, but I think, yeah, there's certainly something that's really changed out there, and it happened much quicker than we otherwise would have thought.
Yes, so yes, we could, and it's quite possible that climate change is contributing to it. But I was interested to determine from both of you as to whether you thought the movement of cattle between countries was a major factor. Certainly, when we look at FMD, there was absolutely no question that movement of cattle was the movement of animals was the cattle, sheep, pigs was very important in the spread across the UK, and the rapidity of spread of LSD virus. I guess I put to you that movement of animals, I would have thought could be a significant factor, but I'd be interested in your views.
Yeah.
Well, I think you're probably right, Michael, and there's so much more of a grey trade than, you know, we could ever appreciate just being here. And that was definitely my experience in Vietnam, and that's, you know, that's how FMD, even in endemic countries, there's been FMD, you know, strain changes and things like that. But at this point, that's really academic, because that horse is kind of bolted. We're almost the last frontier. In terms of, you know, it's already percolated everywhere else. And so, and given we don't bring cattle in, it'll be vectors that affects us.
Yeah. So I agree. I think the other part of that, Michael, and again, I'm not an expert on it, but is the concept that in some of those countries that, you know, cattle are very important, and in terms of economics, from a local point of view, not that they are to us as well. So even if your country has a control program and you know that they're going to confiscate your cattle or kill your cattle and you're not going to be compensated, then the only real option some of those people have is then to sell those illegally in a market where you can't. You smuggle them out and sell them. And so whether that's playing a role, who knows?
Thank you, Tim and Bronte. Question for you, Tim. How do you think regulatory bodies will accept the DIVA system?
Well, I think the classical response to that type of question is that Frankenstein was the doctor, not the monster. But I can't see why it would be a problem. So there would be the same level of safety and efficacy type analysis that we do on current vaccines. And I mean, I guess I'd have to a bit further information on why one would think it would be a Frankenstein virus. But yeah, I can't see there'd be any problem with that. It would be subject to the same regulatory framework that all the other vaccines would be subject to. And then we have to comply with those. So it will be fine.
Thank you, Tim. There's a question here. Was the UK outbreak, and I'm assuming here they're talking about the UK outbreak of FMD in 2001, not caused by dust storms carrying the virus vectors from Northern Africa?
I think that was illegal importation of animal, wasn't it, Michael?
Yes. Well, it was illegal in, well, let's rephrase it. It was almost certainly legal importation of beef from a FMD country, and then the feeding of swill that had not been cooked properly to pigs, that led to what we call a pluming event, where your massive production of FMD virus, and it spread to nearby sheep flocks, and those sheep then, some of those sheep were then taken to a very large market in northern England, the Longtown market, and then went from there throughout the UK and into Ireland, and also into the Netherlands. So that's how we believe the UK outbreak was initiated and spread. Further question to you, and Tim, and also to Bronte. The question is, how do we practically vaccinate in a relatively short period of time the northern herd, given that our primary means of vaccination is via injection? Is there any thought of an alternative to injection?
So we have looked at alternatives in the past, Michael, such as oral delivery, those types of things. And I mean, you're absolutely spot on, as always, in that if the virus comes in during the wet season, if it was to come in via a vector, seems more likely, given we have tropical storms, those sorts of things. Then how do we A, muster, and or B, access, and B, and muster and then access those animals for vaccination? It's a pretty difficult one. Oral delivery is technically feasible, but practically quite challenging. But it's certainly one of the other types of systems that we've been looking at. The only other, we would never be in a situation, I think, where we would consider using theoretically, we could make a vectored vaccine that was transmissible, again, conforming with those the requirements, but I can't see that we would ever actually be releasing that sort of thing. So I think it will probably be driven by, we have to do this, and then we work it out from that. But yeah, do you want to add, Bronte?
Or only that we need to be aware about iatrogenic spread if we had an incursion at the time. So that's another practical issue about vaccinating large groups and needle to needle transfer if you were unaware of an infection, just to complicate things further.
Very, very good point, Bronte. I think that the practicalities of vaccinating the extensively managed beef herd across northern Australia is something that we need to spend more time on assessing how that would actually be done. If we can produce a marker vaccine, then the next question is how do we actually get that northern herd in particular vaccinated, which should then provide a buffer to southern Australia. Final question, because it looks like Kieran's giving us the wind up to finish. Is there anything that we can do with regards the insect vectors to reduce that spread? I'm thinking of some of the strategies that were employed to prevent movement of insects between Mexico and the US. That, you know, there's. Can we look at that?
I think part of the problem there, Michael, as Bonnie alluded to in her talk, was that some of those viruses, they actually infect the insect, replicating the insect, and they're actually amplifying. If one is, I think, the potential breadth of the potential vectors for LSTV, again, make it very challenging. And if they're just in mechanical transfers of the virus, then it's really then the only option is to try and take those numbers down. But if we don't know which of those and vectors are the most important ones, then that can be quite challenging.
Yeah,
Thank you very much.
Certainly, sorry. In an Indonesian context, we have relied on vector control, particularly when vaccine wasn't available or not widely available. And that is not a complete solution, but it can help reduce the spread, the rapidity, the severity of an outbreak. Obviously, the challenge in Australia is that they're not all, in feedlots. So that is a whole nother thing in itself. But that vector control is part of it, but it comes back to vaccination at the end of the day.
Thank you very much, Bronte and Tim. I'll hand over to Kieran to do the wrap-up.
Thanks, Michael, and Tim and Bronte, thanks very much for delivering a wonderful presentation. We had a large number of attendees, so it reflects the interest in this topic and interest in hearing from both of you. So thanks very much. Just to promote next week's presentation as a part of the QAAFI seminar series. It's going to be held at the same time, 12 to 1 on October the 4th. And the title of that seminar will be Soft Particles in Our Food, Instrumental Measurement and Sensory Perception. That will be delivered by Dr. Heather Schrein from the School of Chemical Engineering. So just in closing then, for those people that are interested in getting notifications and regular updates on what's being presented, or general interest in the QAAFI science seminars, this is the contact details listed on your screen now. We would like to invite anyone who has watched it to give feedback and share any thoughts or becoming online more regularly. So just thanks very much for joining today, and we're going to close this seminar off now. Thank you.